Okay, now, so last approach before the next break. It's my pleasure to introduce Ian Skelton, fiber development lead at Astra Bioseparations. And he will talk about addressing process intensification challenges of advanced therapeutics with novel nanofiber absorbent. Stage is yours, sorry.
Thank you, thanks. I'm sorry I put so many long words in the title. Yes, I'd like to talk today. I know we've been talking a lot about cost of goods, and a large part of the product loss through the process is often the downstream process. And, you know, we're thinking about how we can improve those parts of the process by like novel adsorbent development. So Australia is an adsorbent development company. We've got a background in developing ligands, but this is now a completely different set of challenges that we're looking to address with a novel adsorbent. And you see, we're talking about AAV quite a lot today, where, you know, and I think one of the speakers earlier was apologizing for the the low recovery. But if you consider LVV, which we use for like all CAR-T processes, then the recovery from sort of the chromatographic process there can be even lower. And what we wanted to look at was, can we address that with this new technology that we've been developing? And there was also a lot of talk about kind of using currently existing technology, you know, things that we've have served well, the protein purification, the MABs, and that's those technologies, those bead-based resins, they are, you know, they're very fit for purpose for the antibody production process, and to a certain degree for AAV as well. But when we get to larger moieties, when you start talking about things like lentivirus, adenovirus, things like exosomes, you know, they're much, much larger. So the scale of operation in terms of just the physical size of the particle that you're processing becomes a challenge on those kind of traditional adsorbents. And obviously there are current methods that people use to process these things on the smaller scale, like size exclusion or like just kind of physical filtration. But often when we're looking now, as we're getting into sort of more and more characterization of the particles that we use in this therapeutics, those are not necessarily giving us the purity that we need. So what we've done is take an approach to look at how do we process things that are this much larger. And what we've really based our whole approach on is this convective flow media. So this is a media which we have created from two different polymers, and we electrospin those polymers. Now, electrospinning is a process which can be scaled, you know, relatively easily. Particularly over COVID, there was a lot of interest in electrospinning for, you know, masks and fibers of all kinds. And there are some products on the market, you know, that are used currently for downstream processing. But one of the problems with that is the physical stability of the material. And we're addressing that by using two different polymers. We have one polymer that's functionalizable, gives us the same kind of surface that we can put any number of ligands on, as you would with an agarose bead, and another polymer that's there for physical strength and sort of pliability. And that allows us to put this, it basically looks like a sheet of paper or, you know, filter paper. But that extra polymer that we put in there gives it the kind of flexibility we need to put it into any kind of format. The first sort of targets that we addressed with this, you know, we were looking at what are the sweet spots for where we are. So our sort of pore size, effectively, although it's not a pore, it's a sort of path through the membrane, is around 1 micron. The average is about sort of 0.8 micron. So we can look at any of these kind of very large targets, lentivirus, exosomes, you know, large plasmid sequences. Any of those things will easily run through this membrane. And that pathway is not just the sort of largest path through the membrane or through the bed itself. It's pretty representative. So that's 3 different batches made by our kind of process, and then we measure the mean flow perimetry of each of those across a large area of the membrane itself. Imagine a sort of roll of wallpaper is essentially what it looks like. It's just a large roll of paper. And the first target that I'm going to talk about is lentivirus. So, CAR T processes are, obviously in the other track where they're talking about cell therapy. This is a critical intermediate for any of those processes. And there are also in vivo applications as well, which are kind of becoming more and more interesting because the capacity for packaging in a lentiviral particle is much larger than that in an AAV. And they are, the manufacturing process is relatively well established. Again, it's kind of HEC293 cells, and they express these. So the big problem with kind of downstream purification of lenti with the kind of current issues that you see with the current technologies are often around the survival of the lentiviral particle through that process. The reason for the low yields that we see after even primary capture for the lentivirus is that the envelope on the virus is not particularly stable, particularly, for example, in high salts. So when you're using ion exchange processes, you will lose some of the material. And we wanted to address those issues. So if you think of a standard kind of process by which you're isolating your lentivirus, you've got you harvest exactly as you would with the AAV potentially. Nuclease treatment to remove any of the excess host cell DNA and any other kind of DNA complexes you've got there. Clarification, and then AEX. And there are a number of products that people use currently for commercial processes. Resins, but there you've got kind of very long residence times, and you've got very low capacity because your particle can't diffuse into the resin. You've got commercially available membranes and monoliths, but the surface area of those products is a lot lower than the surface area of the fiber, where we've got completely accessible, sort of 360-degree accessibility of the whole surface of the fiber. And we've launched this 2 years ago as a spin filter because we see a lot of people in the LVV space who potentially aren't necessarily familiar at the sort of discovery scale with using FPLC or just chromatography in general. So we presented them with a tool that would be in a familiar format that could be implemented really easily. So you can use that in a centrifuge in the same way that you use like an isolation kit for plasmid DNA. And then last year we launched the 1 and 10 scale units. They have a radial flow geometry, so that allows scalability then up to like much larger scale. The radial flow filters of this kind of design are used in water filtration at enormous scales. And that maintains a bed height that's exactly the same between those two scales and between any scale that we go to. So it's really just a question of the area of this top surface that's accessible that changes. And the nice thing about the little spin filters is that you can do significant amounts of kind of, optimization. If you haven't got the luxury of a computer-aided design to help you out, you have to do the experiments. And we can do that in parallel using the spin filters very quickly. So because we can just load them all manually, put them in as many spots as you've got in your centrifuge, spin everything, wash them, elute them, and look at, for example, different sets of conditions, different transfection conditions, different feed strategies, any kind of set of variables that you want to evaluate for your lenti process. And we're looking here at pretty reasonable recoveries, pretty good process timing. This is all pointing us in the right direction. And obviously this is only worthwhile if you're actually producing clean material at the end of it. And this is a comparison between adherent feed. Often still we see people producing lenti from adherent cells. There are large bioreactors like the Univercells or the Icelis reactors that work with that kind of, in that kind of area. And with that material, actually, the material is so clean, usually, from the adherent expression, that you can just load it directly onto the capture step. It's much, there's a lot lower ratio of impurities in the adherent feed. But even with the more kind of traditional dirty suspension feed, where you've had to, where you do end up with a lot of host cell proteins, host cell DNAs, we still get very good clearance of the impurities. So in that particular case, we're doing one volume of TFF, or just a single volume diafiltration before loading onto the fiber. But that really depends on your your transient transfection method and your kind of your growth strategy, you can actually just load the material directly from clarified harvest onto the fiber. And you can see our capacities there are at sort of mid E9 per mil of adsorbent, and our recovery is sort of 50%. And actually, post-optimization we've been working on more recently, we can actually bump that up to about 80% recovery with some kind of optimization around the elution. And is this scalable? So the answer we hope to show is yes. So this is a 1, a 10, and a 100 sort of mil device, effectively. We've got those. You can come and have a look at them on our booth. They can be seen. They exist. And this is breakthrough curves of just model proteins. So these are all weak anion exchanges. So we're just using BSA, and you can use that to sort of model the charge interaction. The elution peaks there, the reason that the sort of light blue one looks bigger is because that's a 1 mil unit on a 150 mil a minute machine. So that is just a pure sort of diffusion through the system rather than anything else. And you can see the 10 and the 100 there are both run on the same system. So there's always going to be a mismatch between the three. And we wanted to do a comparison with a commercially available Q membrane that's commonly used in sort of primary capture of the lenti. And what we wanted to look at really, the biggest difference that we see is in the actual functional performance of the lenti that we process on this device. So we're looking here at both the sort of measuring the P24, so that's the standard ELISA method that we use to look at quantification of the lenti. But we're also running a functional assay. So the way we do this is transduction into Jurkat cells, and then we look at GFP from our model transgene. And if you sort of look at the light blue line there, that's the lenti-hero, and the breakthrough of actual functional material occurs at a much later volume than for the commercial membrane. And that allows us to basically load more onto our fiber before we see any kind of functional breakthrough. And this is all with VSVG pseudotype and a GFP transgene. We're working on some other pseudotypes at the moment as well, and we do see there are big differences between the charge on different pseudotypes and different sort of growth medium. But the comparison still holds, essentially, between the Q membranes and the lenti-hero membrane. If we also look at where does the functional titer elute, then what we see the biggest difference really between the two. So we elute essentially at a lower salt concentration on the lenti-hero, and we believe that's a function of essentially although there's high capacity and very high surface area, the relative ligand density is actually quite low. So that allows us to elute at lower conductivity. And the fact that it's also a weak anion exchanger versus a strong anion exchanger means you can also play with the pH. Oh, is it going to move? There we go. And the outcome of that is essentially, because the lentivirus is so unstable, or not unstable, it needs to be treated gently. You can't just hold that pool when you elute off an ion exchanger in the high salt. You need to dilute to go down to a stable conductivity. And the sort of primary outcome of being able to elute at lower conductivity is that you then end up with a lower volume to process. So you end up with needing a lower volume or a lower surface area of TFF membrane to do your final diafiltration for your formulation. And that gets more significant as you scale up, obviously. So that's what we believe leads us to sort of talk about process intensification in these kind of processes. Basically, we can use a smaller volume of membrane to load the same amount of material, so that initially leads you to a smaller elution pool, and then you only have to dilute it, but you have to dilute it to a lower volume. So overall, you are shrinking the amount of buffer that you need. You are lowering the volume of final formulation, the final formulation volume, the final formulation area, the filter area. All those things will go down, and that basically allows you to do more, produce more from the same volume of material. And in terms of the sort of scales of operation, you know, we're looking at sort of, it really, obviously, it depends on the titer that you can achieve, you know, with your particular lentivirus. But as a sort of rough guide, you can do around sort of 0.5 litre on the 1 mil unit, about 5 litre on the 10 mil unit. And we're actually seeing some customers who've been using the 10 mil units to cycle, because this was quite a quick process, you can run this up to 10 membrane volumes a minute. So you can run the 10 mil unit at 100 mil a minute. You can process 50 litres on a 10 mil unit, like relatively easily. And we've also now got people moving into the sort of clinical batch space, you know, customers who are already using the 100 mil units to process their 50 litre batches in a sort of single pass. And that's now compatible with CGMP use. So we've produced these units in our clean room, within our quality process. You know, we have sort of 25 years of producing resins for the FDA-approved processes, so we've implemented the same kind of controls on the production of the fiber, and those units are shipped out sort of ready to use. So the other primary kind of raw material that people are using for almost in fact all real processes using ATMPs is plasmid DNA. So the second sort of set of data I'd like to show is relating to processing these kind of sequences. And the nice thing about this material, again, is that we've got this very broad flow path within it. So we have very low pressure operation, even at high flow rates. So, you know, we've talked a lot about AAV and the different plasmids for the different transfection processes. A standard AAV helper is between 11 and 15 kb. So that immediately makes it really quite large to process on standard adsorbents. And as we've seen, you know, through looking at different the different kinds of nucleic acid that are required, you know, either as the payload or as kind of as you desire for compressing multiple sequences into a single plasmid, you know, that then the length of pDNA that is required from a single expression is becoming larger and larger. And that's why we have our pDNA hero. It's a weakly hydrophobic surface. This is just a set of transition curves, you know, as you'd use to characterize a column, rather than looking at HETP, because the bed is millimeters thick. You can't really look at a pulse. We do it with a salt transition curve, and that is a 1, a 10, and a 100 mil unit normalized to column volumes, showing that we can overlay that, so you can scale this process up to the 100 mil unit. And we've looked at this to use to purify sort of very large sequences. So we're looking at that's almost a 35 kb sequence. And you can see the pressure trace there. That's all running at 5 mil a minute on a 1 mil unit, just on a standard FPLC. And the pressure there, the delta column pressure, is never getting anywhere near. It's less than half a bar throughout the entire process, all the way through the load and the elution. And this is a fairly standard process that we use. We just, at the moment, with this, we were doing calcium chloride to remove a load of the RNA. And then we're just diluting that with ammonium sulfate and binding that lysate directly onto the fiber. So we've looked at this and scaled this between the pDNA Hero 1 and the 100. This is an AAV helper plasmid, so the kind of thing that a lot of CDMOs will produce off the shelf, just as a standard intermediate or a raw material to go into a process. And we've got there the traces from the 1 and the 100. The 1 is run on an FPLC, and 1 is run on a system where we're running it using a peristaltic pump and a set of kind of disposable transducers to measure the pressure and the UV and the conductivity. But we're running that 5 mil a minute on the 1 mil, 500 mil a minute on the 100 mil. And you actually see, because you can reduce the dead volume effectively in the system that you make yourself, we can cut down the runtime quite significantly. So we're running around 8 liters of lysate on that in 40 minutes. And this is a gel just showing the kind of comparison between the two. So we're recovering the material in roughly the same sort of ratio. The reason the recovery is a bit low there, we're looking at total nucleic acids in this, so we're measuring it from, we've got some, this is from the lysate, so it's more difficult to accurately sort of estimate the concentration at the beginning. So that's one way of using the material, just as a primary capture, and we're working on some even more efficient ways of removing the RNA that we will be sharing hopefully soon. But those processes are just using a standard calcium chloride precipitation. But the other way that we use the hydrophobic surface, often in a lot of standard processes, you'll do ion exchange capture and then hydrophobic polish, as an orthogonal step, and remove all the final impurities. And this is the standard way of making CGMP grade plasmid sequences. And when we use this as a polish step, so we use this as a step after capturing the sequence from the lysate on our resin, so we can pack a very large column with the DEA resin. And in this case, I think we just processed a large amount, as large a column as we could run on our actor in the lab. So this we can scale this to whatever size we have, but we only have a 150 milliminute system in the lab. So we processed this, and then we did a equal volumetric load onto a 1 mil and a 10 mil pDNA hero, running the 1 mil at 5 mil and the 10 mil at 50 mil. And you can see the profiles are very similar. We're just running a linear gradient from 3 molar ammonium sulfate down to just TE. And the profiles are very similar, and the recovery is very similar. So here we're getting over 80% step recovery from there, and the material is very clean. And the unit itself, the material is very stable. So the ligand that we're using is essentially completely stable to sodium hydroxide, and the fiber itself is produced in a process which involves 1 molar sodium hydroxide. And we can this experiment is ongoing. We expect to be able to run far more cycles for this, because the material that we're loading on is very clean. It's coming from it's already been through primary capture. And in terms of the COP process on this, we do, I think, about sort of in total, it's around sort of 10 minutes of total contact time at high pH. And the whole run is around 30 minutes to process, to give us 2 mg, 2.15 mg every cycle. And this will scale up to whatever size we can produce. So that is a picture of the actual unit itself. So we've got it's set up so that you can just have it stand on the bench. Doesn't need any kind of external press or holder or anything like that. It will be compatible with, say, using on any of the disposable chromatography systems that are available, sort of the Acta Ready or the Repligen systems. And we used that with the Centre for Process Innovation over in the UK, because obviously our operation is limited by the scale that we can produce material at. But they produce for us an 8 kb plasmid, an M-cherry plasmid, in standard sort of E. coli expression, at 100 litres, so that we could have a industrially relevant feed to demonstrate the 1 and 100 scale pDNA heroes. And that was done after a primary capture using our DAE pure bead resin. At the lab scale, we did that on a 50 mil pre-packed column. That's a plastic column that we can supply pre-packed with 10 centimetre bed height. And then for the large scale, the pilot scale run, the CPI packed a column with the same resin at about 120 millimetre bed height. Again, this was limited by the size of skid that they had to use. And overall, the kind of time taken to process the whole set of for a single pass through the entire process, for the two-step process, the chromatography on the pDNA hero, it takes basically the least amount of time. So if you're looking at a standard resin process where you might have to run at kind of 3 to 6 minutes residence time, you'd be looking at several hours for that. You'd be adding on for every polish step that you add. And we're getting, it's a standard alkaline lysate, calcium chloride precipitation, filtering that through a depth filter, and then UFDF to go into the primary capture step, removing most of the RNA in that on that first step on the DEAE, and then we're doing the final polish on the HIC. So that's a comparison of the traces for the pDNA Hero 1 and 100. In this case, this was run on the pDNA Hero 1 is run on a standard FPLC, like a 25mm machine. And then the Hero 100 is run on a pilot system. The pressure from the system was limiting in this case, so we couldn't run it to as fast as we wanted to. You could actually run this at 1 liter a minute if you had a system that would go that fast. And we were able to recover 73% from that from the 1 mil unit, 75% from the 100 mil unit. And the purity of the material is very comparable. So you've got here the amount of open circular at each step, at each scale, and then the supercoiled. This is all measured by HPLC. Usually we're looking for around 95% for the supercoiled pDNA. But in this particular case, we actually had quite a lot of, the major impurity was a multimer. And that's something that we've been talking to some customers that they see that with certain sequences that is quite challenging to clear. And we're also clearing quite a lot of that as well as the open circular material. And you can see that the pDNA Hero 1 and the pDNA Hero 100 are both giving very similar performance in terms of purity of the nucleic acids produced. And in terms of clearance of the other impurities, if we look at the FDA guidelines, we are hitting those with that pDNA Hero 100. So we're removing all of the endotoxin in that essentially, or as much as we can measure. And then we're hitting the specifications for the host cell DNA and host cell protein. So when we think about kind of scaling this up, or the kind of scales of operation that we're looking at with the currently available products that we're launching now, so we can produce 1 gram of purified material in a single shift, on the pDNA Hero 100. And if you're looking at sort of small scale, sort of scale down production or process characterization, then the pDNA Hero 1, will give you your MIGs of purified plasmid. And that correlates to various sizes of the pre-packed columns that we have. At the smaller scale, you could use, say, like 3 of the little 5 mil columns, almost sort of screwed together. And then we have a whole series of pre-packed GMP grade columns that we can supply with the PuraBead, with the DEA. And they are available in a number of different sort of diameters and bed heights, all the way up to very large scale process scale columns, which we can also pack. And they are all made, under our quality process, packed in the clean room and released with bioburden and endotoxin, as you'd expect from a kind of CGMP grade column. So yes, that's the end of that. And again, there is no timer, so I don't know if I'm time for anything. So that's good, right? So yes, any questions?